We're very happy to have you here, and it really was a great symposium. Every 2 years, we do this every 2 years. It's always different. There are always different things that come up as the most exciting. We tried some new things this time. We had a pre-conference to help the basic scientists learn how to start doing research on people as opposed to just rats, and that was quite well attended and very good. We had some great international basic scientists here, most of whom are not still here, but who really presented some great data. And then we challenged the participants. In the past, we've given out pilot grants of $10,000 to $20,000 to sort of jumpstart people's research. And this year, I decided to do it a little differently, and instead of taking them ahead of time, we actually had the people on site during the conference develop consortium that had to include a basic scientist, a clinician, and an advocate, and then answer some of the basic questions. And it did, it worked. We got 9 applications, and you'll hear the winners tonight at the dinner. But the other thing it did, which the unintended consequence, which really pleased me a lot, was it actually made people talk to each other outside of their silos. Because since you had to have a basic scientist and a clinician and an advocate, all of a sudden you had basic scientists trying to find an advocate when maybe they wouldn't have talked to them otherwise, or clinicians looking for a scientist. So it really worked at increasing the conversation at lunch and at breaks in order to put forward these consortiums. So I think it's one of I try new things, and some of them I keep and some of them I don't, but I think that one's a keeper. I think it does some great stuff, and we'll see what the results are. So what I'd like to do is go down the panel. We have several people who've been here for the whole conference, and really get them to give a little bit about what they thought. And most of them have been to this conference before, multiple times actually, and what were the highlights for them at this particular symposium, and you know, what new ideas and where it's going to take them. So we'll start with Dr. Seema Khan, who is a breast cancer surgeon from Northwestern University and an excellent breast researcher.
So this conference didn't disappoint, as with all the others. I think the unique aspect of this Intraductal conference is that it brings together people who wouldn't necessarily have met in the usual settings, in the mainstream conferences that we go to. And there are always people with data that's a little bit edgy, so I guess that yeah, sort of goes along. It's part of the theme. But it really is very provocative usually, and makes one think with a different perspective and come up with different solutions. So I heard some very nice summaries of progress that has occurred in different areas of research over the past few years. The one that I was particularly taken with was Dr. Catherine Briskin, who talked about the way hormones affect normal breast cells. It's an area that I was very interested in when I began my career. I've sort of drifted away from it a little bit, but it was fascinating to hear the whole story as it's developed since, and then to take those developments. For instance, she described some of her work highlighting the importance of progesterone in the normal breast physiology and the development of breast cancer, which is something that I've been thinking about a lot recently as well, and sort of helped me crystallize some of these ideas. And then the other part of it is meeting people you wouldn't otherwise have met. So I've been trying to reach Dr. Ron Wheely, for instance, for the last 2 months or so, and we haven't really connected, and he's here, and so I was able to have a really nice in-depth conversation with him that has sort of prepared the way for us to go forward in terms of the collaboration. I connected with other people I've been trying to reach that helped me solve problems I've been facing in the work that we do, and so on. So it's really a very, very fertile environment where all sorts of ideas come up and can be developed, and hopefully will be taken forward once we leave here. So I really enjoyed it.
Good. And Dr. Sheldon Feldman is from Columbia University in New York, and one of our collaborators, or potentially, we have a grant in with where we're going to work together, and he's also a breast surgeon.
So this meeting really is wonderfully rich, and as Seema was saying, Dr. Khan, it is a great opportunity to interact with other people who are like-minded, who are in different disciplines, though, who are not clinicians, who are basic scientists, and we learn a lot from each other. I think that the two areas that I think many of us have been trying to move forward has been the idea of a better breast screening test, looking for biomarkers in nipple fluid, something that would be easily obtainable so that we can really evaluate patients in a minimally invasive way. And I think there is progress described at this meeting about proteomics and microRNAs, looking at nipple aspirate fluid, which I think is very promising. And there's, out of the collaborations, I think there'll be some new trials which will come out, which will further inform this. So I think that's huge. We've also, a number of us have really been excited about the idea of minimally invasive approaches to treating breast cancer intraductally. And there's been some early clinical work done, and I think there's some new information that perhaps we have to make a slight shift in this direction in terms of the agents that we're choosing to use, more targeted to perhaps the disease specifically. In the same way, some of the instruments that we can apply to access the breast through the nipple, ductoscopic equipment, we see continuing to advance with new technology, better resolution, more user-friendly. So these are all very, very encouraging. So I think that come away from a meeting like this feeling enthused and having learned a great deal, and that we are truly making progress, and that a lot of the concepts that Susan has put forward in terms of intraductal approach really are coming to pass, maybe a little bit too slowly, but that we're making progress. So it's a very, very great meeting.
Next, one of the things we're very strong on in this meeting is we give out scholarships to the advocates. We have at least 5 advocate scholarships, and we also have advocate speakers. And Sandy Walsh is a breast cancer advocate who I've known for a long time, and they did a panel for the research scientists on how to actually use advocates in your research. A lot of the grant applications these days require that you have an advocate, but the scientists don't know what to do with them. And so Sandy was part of a great panel telling them all the different things, and that they actually, just because they had breast cancer doesn't mean they don't have a brain, and that they actually can contribute to the science as well as patient recruitment. Sandy, what was your takeaway from the meeting?
Yeah, the panel was fun, trying to tell them that there really are ways that advocates can be trained and can understand what's going on. And it was exciting to see so many advocates here, and the communication that was going on because of the grants. And the other part that I really liked is seeing the clinicians and the basic scientists, the epidemiologists, getting together and working together. That's so exciting. I'm a part of the National Breast Cancer Coalition, and we have a new agenda called Deadline 2020 to end breast cancer by 2020. And this is one of our goals, is to cross these silos, get different people talking to each other, get new ideas. And so this conference was really exciting in that it was making big strides towards doing that. Want me to introduce myself?
No, I'm waiting for you to move your thing over. This is Dr. Ellen Mahoney, who's also, we're high on breast surgeons right now, but as we said, we can answer all kinds of questions. We're pluripotent, surgeons to quote Thea. And Ellen was at Stanford and now is up in Humboldt County, and was one of our collaborators on an intraductal study. Also has been to, I think, most, if not all, of these intraductal conferences, and as a breast surgeon.
Thank you. There is not another conference where basic scientists, clinicians, and advocates are actually encouraged to speak to each other. I think everyone, including San Antonio, which is giant, or ASCO, has all of those components there, but nobody makes any attempt to actually speak in a language which the others can understand. And here, not only can the others understand it, but we've all been working toward a particular goal through all of this series of conferences, and that's to understand how the breast is put together And it's astonishing to most people to realize that the inside of the female breast is the last unexplored area of human anatomy. It's not like it's not a public health problem, but somehow nobody thought it was important to figure out how many ducts there are, how they're arranged, how they work together. And when we started to figure that out, it's been surprisingly hard, and probably why it hasn't been solved. But really, until this organization and this movement started, no one was really looking at it very hard. They were looking at cells, but they weren't looking at the cells as a pattern. So, and then in the office, and any of you who've had breast cancer understand that when we do surgery, we treat the breast as if it's one hunk of clay, and it's our job to take out a piece of that clay, and we pay no attention to the inside anatomy either. But if you think about the breast, a very small part of it are actually the cells that can become breast cancer. The rest of it is not quite an innocent bystander, but we don't understand its role. But again, when we do surgery, we take out a lot of things that we don't need to take away. And we would like to be more selective and go right to the heart of the cells that are the targets for whatever is causing this. So every time we come to this meeting, we get a little closer. About half of the people come back every time, and we've gotten to be good friends over the years, and the other half we're refertilized and re-energized by new people who come in, and it's just been a wonderful experience. It's the best meeting I attend.